The role of bioluminescence in studying circadian rhythms. Title: The role of bioluminescence in studying circadian rhythms. Introduction. Circadian rhythms refer to the biological processes that follow a roughly 24-hour cycle and regulate various physiological and behavioral functions in organisms. These rhythms are found in almost all living organisms, from plants to animals, governing essential processes such as sleep-wake cycles, hormone regulation, and metabolism. Understanding the intricacies of circadian rhythms is crucial, as disruptions to these biological clocks can lead to a range of health-related issues. Bioluminescence, the emission of light by living organisms, has emerged as a powerful tool in studying circadia and rhythms. In this article, we will explore how bioluminescence has revolutionized our understanding of these rhythmic patterns and its contributions to this fascinating field of research. Bioluminescence and the circadian clock. Bioluminescence is a phenomenon in which living organisms produce and emit light through specific chemical reactions. Bioluminescence is tightly regulated by circadian clocks in these organisms, which orchestrate the production of light in a rhythmic manner. The use of bioluminescent organisms, especially those with reporter genes linked to clock genes, has enabled researchers to monitor and visualize the underlying mechanisms of circadian rhythms. The discovery of the green fluorescent protein, GFP, and its subsequent genetic engineering paved the way for real-time monitoring of clock gene expression through bioluminescence. The circadian clock and gene expression. The molecular regulation of circadian rhythms is primarily achieved through a complex network of clock genes in a feedback loop. These genes, including PER, period, CRY, cryptochrome, and BOL1, BR, AIN and muscle aren't like protein 1, among others, undergo rhythmic expression throughout the day. The expression of these genes influences various physiological processes, ultimately driving circadian and behaviors. By employing bioluminescence as a tool, researchers can fuse these clock genes with reporters such as luciferase. Luciferase is an enzyme that catalyzes light-emitting reactions involving luciferin, producing bioluminescence as a result. When clock gene promoters are fused with luciferase and introduced into cells or organisms, the bioluminescence emitted allows for real-time monitoring of gene expression. This technique, known as bioluminescent imaging or real-time bioluminescent recording, has revolutionized the understanding of gene expression patterns in circadian clocks. Understanding the synchronization of circadian clocks. Bioluminescence imaging has also shed light on the synchronization of circadian clocks at the cellular and organismal levels. Synchronization is essential for the proper functioning of circadian rhythms and ensures coordination between different tissues and cells within an organism. By measuring the phases of individual cellular clocks in a population, researchers can analyze how synchronization occurs. Bioluminescent imaging techniques have revealed intricate intercellular communication and fine-tuning of oscillatory dynamics for proper synchronization. Bioluminescence and advancements in chronobiology. The application of bioluminescent techniques has accelerated advancements in chronobiology, the study of biological rhythms. Being non-invasive and highly sensitive, bioluminescence imaging has allowed scientists to explore various aspects of circadian rhythms that were previously challenging to investigate. Bioluminescence has enabled the visualization of clock gene expression in specific tissues or cells within living organisms, providing a spatial understanding of circadian timing. This spatial resolution tie-in has proven invaluable in deciphering how different tissues maintain synchronized rhythmicity, especially given their varying needs and environments. Additionally, real-time bioluminescent record ing has allowed researchers to explore the effects of genetic and environmental perturbations on circadian rhythms with high precision. Future perspectives. The utilization of bioluminescence in studying circadian rhythms has undoubtedly enhanced our understanding of this field. Bioluminescence has revolutionized the study of circadian rhythms by providing valuable insights into the underlying mechanisms and dynamics of these rhythmic patterns. Through the use of bioluminescence and organisms, such as algae, fish, bacteria, and fireflies, researchers have been able to monitor and visualize clock gene expression in real time. The molecular regulation of circadian rhythms primarily involves a complex network of clock genes that undergo rhythmic expression throughout the day. By fusing these clock genes with luciferase, an enzyme that produces bioluminescence, researchers can observe gene expression patterns in real time. Furthermore, bioluminescent imaging has provided insights into the synchronization of circadian clocks at the cellular and organismal levels. Synchronization is crucial for proper circadian rhythm functioning and is achieved through the adjustment of clocks to external cues, such as light and temperature. These findings have implications for understanding diseases associated with disrupted circadian rhythms, including sleep disorders and metabolic dysfunctions. Advancements in bioluminescence imaging techniques have also accelerated the field of chronobiology. Additionally, real-time bioluminescent recording has allowed for the precise investigation of the effects of genetic and environmental perturbations on circadian rhythms. Bioluminescence has opened up new avenues for studying circadian rhythms, offering researchers the ability to monitor and visualize clock gene expression in real time. This has been made possible through the use of bioluminescent organisms, such as algae, fish, bacteria, and fireflies, which exhibit rhythmic production of light. By genetically engineering reporter genes like green fluorescent protein, GFP, to be linked to clock genes, scientists can track the expression levels of these genes over extended periods. Understanding the molecular regulation of circadian rhythms relies on identifying the complex network of clock genes involved. These genes, including PER, CRY, and BUL1, undergo rhythmic expression throughout the day, influencing various physiological processes and driving circadian behaviors. By fusing these clock genes with luciferase, an enzyme that produces bioluminescence, researchers can visualize the real-time gene expression patterns. This technique, known as bioluminescent imaging or real-time bioluminescent recording, has revolutionized our understanding of gene expression dynamics. CS and circadian clocks. In addition to studying gene expression patterns, bioluminescence imaging has shed light on the synchronization of circadian clocks. Proper synchronization is essential for the coordinated function of circadian rhythms within an organism. Through the use of bioluminescent reporters, researchers have been able to investigate how clocks adjust to external cues, such as light and temperature, to align with a 24-hour day. By measuring the phases of individual cellular clocks in a population, scientists can analyze how synchronization occurs and uncover the intricate intercellular communication tie-in involved. Advancements in bioluminescence imaging techniques have propelled the field of chronobiology forward. Being non-invasive and highly sensitive, bioluminescent imaging allows scientists to visualize gene expression in specific tissues or cells within living organisms. This has provided a spatial understanding of circadian timing and how different tissues maintain synchronized rhythmicity day to their varying needs and environments. Moreover, real-time bioluminescent recording has facilitated the precise investigation of the effects of genetic and environmental perturbations on circadia and rhythms. Looking ahead, the combination of bioluminescence techniques with genetic engineering tools like CRISPR-Cas9 holds immense potential for further unraveling the complexities of circadian clocks. Thanks for watching.